Okay, good afternoon, everyone, and welcome to today's Psychology, Neuroscience, and Behavior Colloquium. It's always a special day for any faculty member when one of their students is preparing to defend, and today's colloquium by Lucas Greville is no exception, because it signifies that he is preparing to defend his doctoral dissertation later this summer. Of course, it's a very important achievement. It's actually almost 2 years ago, just only about 10 days off the mark, that my last PhD student gave a colloquium in this very room. For those of you who don't know, we should actually go on. Who is Lucas, James, Steve, and even though I guess it's Stefan, but Greville. Lucas is a native Ontarian, born and raised in Toronto. And yes, he is a true blue die-hard Maple Leafs fan, in case some of you didn't know that already. Lucas completed his undergraduate degree at McMaster in the honors of biology and psychology, biology and psychology, neuroscience and behavior double degree major. It's a very rigorous program. It actually has our highest entrance average to get in all the PNB programs. Over the years, Lucas has earned many academic awards and honors. He was on the Dean's Honor Roll every year at McMaster, got an OGS award for his first year of his master's, got an NSERC award for his second year, got an OGS for his first year of his PhD, and got an NSERC CGSM for his doctoral degree. And also recently got a MyTax Global Link Research Award for travel to Berkeley this last summer to do some collaborative work with our collaborator, George Bentley, who will be giving a colloquium, hopefully, here next month. So Lucas, in addition to having this great scholarship and academic honors, he also has a few publications that he's produced since coming to McMaster. Not all from my lab, not all with Dennis Catanzarada, who has been Lucas's co-supervisor for his bachelor's honors, for his master's, and we're going can say, for his PhD, even though Dennis technically is emeritus right now. So I first met Lucas. And I expect there'll be a couple more papers from some of the work that we're going to hear about today, because he's actually not going to talk about all the published work. I first met Lucas when he was a sophomore biopsych student in the spring of 2012 at one of our famous undergraduate hosted Meet the Professors night over at 1280, I guess it was. And despite actually meeting and talking with me that night, Lucas decided to come to my lab and pursue a summer research position with me. This is what Lucas looked like back then. This is at one of our early lab sterilization parties. I won't tell you what was being sterilized, but anyways, Lucas was and still is a tireless leader, volunteer, and role model. Yes, he was literally a drum major and in the McMaster marching band and helped get the new uniforms, as he was part of the group to get the new uniforms for McMaster's marching band. And he is a leader not only in the band, but he's a leader amongst our graduate students by his academic and research work. He's also very integrated and networked socially with the department, and I think a lot of the grad students know that. So Lucas, still looking really young, working with me with some bats in the lab, not afraid to do some of the hard work, getting up on the ladder, looking like city workers, waiting for something to happen. There he is again, with Ryan and Paige, a couple of other undergraduates from many years ago. Lucas started to develop his own personality in the bat lab, started to worked also closely with other undergraduates and graduate students. Here's one from my last PhD student Roberto. Roberto has changed weight over the years, but Lucas has not. In fact, we encourage Lucas to eat whatever we pick him up. Here's another photo just of a young Lucas in the Toronto Zoo holding an Egyptian fruit bat. And this is also a picture quite some time ago when Lucas was in his mullet phase, working with Joel James. All right, so I want to talk a little bit about Lucas's research. Lucas studies mammalian steroid hormones. His work focuses on how hormones are absorbed in the mammalian tissues, how they are transferred between conspecifics, where the hormones bind in normal tissues, and the natural cycle of hormone production. And he studies this in an uncommon mammal model, the big brown bat. And actually, Lucas's work has been pioneering. He's done the first demonstration of hormones as a pheromone in bats, period, along with Dennis DeCatanzaro and Tyler Pollock in the initial paper that formed Lucas's bachelor's thesis. Now, at some point, I started to worry a little bit about Lucas, that his work with hormones was getting a little too serious, and maybe he was getting a little too close to his chemicals in the lab, because he literally transformed from, this quite young-looking undergraduate into quite a massive, almost overnight. It seemed like within weeks. I came from a professional model, as you can see here, he's airbrushed up nicely, and a professional lumberjack overnight. But seriously, Lucas has been an absolutely wonderful student. In fact, he enjoys this photo so much that he decided to wear the exact same shirt today for today's talk. It's been a pleasure watching Lucas develop his technical research, writing, and mentoring skills. And Lucas continues to be a good mentor. You can see he is helping Roberto in his work. And Lucas always aims high in his research and is a solid team player who really enjoys helping others in the lab and also helping others in his community. And so without further ado, I'd like to introduce Lucas Greville, who will We will speak to you today on examining the pheromone potential of estradiol in the big brown bat. We're also debuting our brand new lab logo, which I'm sure the McMaster logo police won't like.
All right. Thank you, Paul. Thank you, everyone, for coming today. As everyone I think, knows, and as Paul's alluded to, I've been in the lab for quite a long time. So I've worked with Paul for quite a long time. And not quite this long. I've never been around for the beard days of Paul. But as Paul did say, as an undergrad, I think I am now one of the only graduate students who worked with Paul when he did still have hair, which was back many years ago. And this was one of our first bat catching experiences where I don't think we actually caught any bats because I don't think we deemed the roof safe to climb on at the house, but we did get to play with some cute kittens, which was good. And when I came into the lab many years ago, Paul promised me a wide array of scientific experiences, which he has definitely delivered on. But that shouldn't surprise anyone who knows Paul, because Paul is a man of his word when he's forced to be. And throughout my experience in the lab, though, Paul has delivered on that promise, and I have had a wide array of experiences. Paul's also been a big proponent in graduate students having a life outside of academia. For me, that's largely sports. I try not to take quite after his athletic abilities, but Paul always does focus on the big picture, which is to keep your eye on the end goal or the rock, no matter where your body is lying on the playing surface or how many limbs you have in contact with that playing surface. But as you can see today, and for those who know Paul over a longer duration, he's also had a change in style. Thankfully, I think we can all be grateful that his style has changed since his early research days many, many years ago. Although I do like these shoes, they're kind of stylish, coming back in the trend. But Paul's always been very passionate about research as long as I've been in the lab. Whenever I came to him with a new research idea or with data, you can see that passion and excitement for research that Paul has, and he really instills that on his students. So thank you very much, Paul. And what I'm going to be talking to you today, as Paul alluded to, is my dissertation work examining the pheromonal potential of estradiol in the big brown bat. So kind of to get into the work here, I want everyone to kind of imagine an interaction between different organisms. It can be any mammal, any type of interaction. And the interaction you're probably picturing in your head is something similar to that in which researchers typically focus on. Something probably to do with physical contact between individuals. It might be grooming, it might be playing behavior, it might be mating behavior, but something deeming between physical contact. Other types of interactions include auditory or vocal communications, as well as visual displays, body language between individuals, or colorful displays that signal information. However, there's another aspect of animal interactions that individuals often ignore, and that is that of the chemical signaling between individuals. And often this comes in the form of a pheromone. That's a substance that's excreted by an individual into the environment that alters other individuals' behavior or physiology. So to give you a couple examples, we have ladybugs, which secrete aggregation pheromones and cause other ladybugs to come into the same general vicinity. We have alarm pheromones. Not sure what button I hit there. There we go. We have alarm pheromones in yellow jackets. So when a yellow jacket is injured, they'll release an alarm pheromone so that other individuals know that there is danger present in the area. We have foraging or trail-making pheromones in army ants that allow the colony to get more efficiently to a food source and back to the colony. And anyone that has a male dog is probably more than familiar with territory marking in the species, in a variety of dog species. And when we focus more specifically on sex pheromones, we see that these occur across a wide range of taxa as well. So for example, sea urchins release a pheromone along with their sex cells to signal for other sea urchins in the area to also be releasing their sex cells at the same time. This is how these animals reproduce. Tracking pheromones in moths released by female moths allow males from kilometers away to track and find the females for reproduction. There are pheromones in the tear fluids of male mice that elicit reproductive behavior and lordosis positioning in female mice. And in pigs, boars have androstenone in their saliva, which signal for estrus females or females that are in the reproductive phase to go in and start to present sexually receptive behaviors. Now, if we look at more behavioral phenomena, due to the introduction of novel individuals to the environment, We have a number of behavioral studies that are shown across a wide range of taxa in mammals. These studies have been shown, and these effects and studies have been shown in voles, gerbils, sheep, wild and domestic horses, galadas, but are best studied using the mouse model. So I'm going to be using the mouse model as I take you through some of these behavioral effects. And the first of these effects is the Bruce effect. This is when we introduce a novel adult male to an environment, to a female that is either inseminated or perhaps in the early stages of pregnancy. And when we introduce this novel male to the female's environment, we find that she has a successful pregnancy at a significantly lower rate than if the male was not introduced to the environment. So we have a disruption of pregnancy due to the male's presence. Another similar effect is the Vandenberg effect, and this occurs when we have juvenile females of a species introduce a novel male to the environment, and these females undergo precocious puberty or reach sexual maturation at a younger age than they would if the male was not present. So in both of these effects, you can understand how, from the male perspective, he's able to increase his own reproductive fitness, because in this case, the male is now able to mate with females that otherwise would not be sexually mature. And in the case of the Bruce effect, he is able to mate with a female once she goes back into heat, and she is therefore going to sire his offspring as opposed to the offspring of another individual. I also want to point out that the introduction of males to environment is also enough to elicit sexual behavior in females, as well as ovulation in a number of species. And these effects are well known and well established in the mammalian literature, but the large question as to how they occur remained for a long time. And a lot of work from Dennis de Catanzaro's lab throughout the 2000s has shown that estradiol is a major player in stimulating these effects. So what is estradiol? It's a sex hormone found in all mammals. It is fat soluble with a low molecular mass, and it is very stable. It's highly considered the most potent sex steroid, as well as the most potent hormone in the mammalian endocrine system. Estradiol is so potent, in fact, that simply placing small quantities of estradiol in the nasal membrane and nasal region of female mice is enough to stimulate and induce both the Vandenberg and Bruce effect in mice. So prior, I was talking about the induction of these effects when we introduce a male to the environment. So this is great that estradiol does it, but where is this estradiol coming from? More work in the de Catanzaro lab showed that male urine contains enough estradiol that simply placing urine from a male on the nasal regions of a female will also induce both the Bruce and Vandenberg effects. Behaviorally, when we separate males from females, and this might be an inseminated female, it could be a juvenile female, and we separate them with a wire mesh grid, male mice will direct their urine towards these females. And this is in opposition to typical male behavior of urinary patterns, and where males often put droplets around the environment as more of a territorial marking process. So here we have a change in male behavior. Now, what's even cooler is that we're using radioactive labeling. Dennis's lab has shown repeatedly that male mice transfer radioactive hormones to female conspecifics. So the hormones are actually transferring from the males through the urine to the females, which is awesome and super cool, in my opinion. So I said, in order to trace these hormones, you have to use radioactive hormones. So why are we using radioactive hormones in such studies, and what is the purpose of this? So typically, the lab uses tritium, which is a form of hydrogen, and we're able to purchase estradiol that is labeled with radioactivity with tritium, so it's tritiated labeled estradiol. And this allows us to measure the levels of radiation in the tissues of individuals, with that level of radiation or radioactivity corresponding to the levels of transferred estradiol in such experiments. So we know that this estradiol has transferred from male to female individuals, It isn't just an upregulation in estradiol present in the female due to the male's presence. And of course, we're able to correct for background radiation. Now, what's important to note when using radioactive estradiol in these experiments is that it is in fact bioactive. So what this data here is showing, on the y-axis, we have the radioactivity measurements in DPM, or disintegrations per minute, per milligram of tissue, and a variety of tissues sampled on the x-axis. And in this project, we have an individual who has been injected with radioactive estradiol paired with a conspecific female. And that female is where the radioactivity in this diagram is pertaining to. So first, if you draw your attention to the black bars, these is a level of radioactivity found in an individual who is paired with a stimulus individual that had been injected with radioactive estradiol. And what we see is that it transfers to this conspecific at a specific rate. Now, if this conspecific before being paired is injected with just normal, unlabeled estradiol, in the gray bars here, we see a decrease in the amount of radioactivity measured in that conspecific's tissue, suggesting that both the bioactive estradiol in that conspecific as well as the transferred radio-labeled estradiol are competing for the same receptors in the conspecific's tissues. So because the tritiated estradiol and the unlabeled estradiol compete for the same receptors, they are both bioactive. So we know that this hormone that we're injecting the animals is indeed bioactive and can bind to the same receptors as normal estradiol. So as Paul alluded to, during my undergraduate thesis, when I joined the lab, is when we started these collaborative projects with Dennis's lab. We set out to look at this potential of hormone transfer in bats. Now, the Vandenberg and Bruce effect have not actually been shown in a bat species. So we were setting out to really look at whether the potential for it to occur is there, whether the potential for estradiol transfer in bats is there. And first off, we had to make sure that female bats are able to absorb estradiol from the environment. So in order to do this, we exposed female bats to 10 microcuries of tritiated estradiol. So microcuries are a measurement of radioactivity in which we standardize our doses amongst individuals. And we exposed the females either on their abdomen or intranasally with tritiated estradiol. And 1 hour later, we found that it was present in all of the neural, peripheral, and reproductive tissues of these females. So female bats are able to absorb exogenous steroids from their environment. To look at whether males will transfer estradiol to females, we injected a bat with 50 microcuries of estradiol. And we paired them for 48 hours with 3 female conspecifics during the mating season of the bats. And what we see is that we measure radioactivity in all 3 females this bat was exposed to. Somewhat idiosyncratically, however, transfer did occur to every female. And we replicated this multiple times. So what I'm showing you here is one replicate of this project. We replicated this multiple times with male bats transferring estradiol to both adult and juvenile females. So yes, bats are able to transfer hormones from male individuals to females. So this is the work that I've really started to elaborate on and continue throughout my PhD. And for all of these studies, the model organism we're using is the big brown bat. So at McMaster, we're lucky enough to have Canada's only captive research colony of bats. All of our bats in our colony are either wild caught and brought into the lab, or the animals are bred in captivity. The big brown bat is found across Canada and throughout North America, south towards the northern tip of South America. It's an insectivorous species, typically weighing about 14.5 to 18 grams in the wild. And our animals get a little girthier than that in the colony, but we do expose them to environmental cues, including natural lighting. The temperature fluctuates with the ambient environment, and animals are able to fly and move around the colony, and they have a number of places in which they're able to roost, such as under these towels here. So you're probably wondering, why bats? Why are we going to look at this hormone transfer in bats of all animals? There's a few reasons for this. As some of you may not know, I've probably instilled this thought on you many times already. Bats are not rodents. Mice and bats are evolutionarily quite distinct, believed to have formed different taxa approximately 92 million years ago. The oldest bat fossil we have on record is approximately 52 million years ago. So although the Germans referred to the bat as Fledermaus, the fluttering mouse, bats are not rodents and they are not mice. So by looking at hormone transfer in a new novel species that is evolutionarily distinct from other organisms in which hormone transfer has been documented, we are able to perhaps predict that this might be a mammalian phenomena as opposed to just a rodent phenomena. The second is the roosting behavior of the bats. So when we're looking at hormone transfer, you need animals to be in close proximity to each other. This picture here obviously shows a number of individuals roosting close in proximity to other individuals in very close confines. This is a larger cage that the animals have decided to all roost together as opposed to spread out throughout the cage. So when we're looking at hormone transfer potentially through excretions, this is a prime situation in which individuals do not move when they're going to be defecating or urinating, and they'll just relieve themselves, and whoever's in the vicinity is in the vicinity, and that creates ample opportunity for hormone transfer to occur. The last reason is the maternal harem structure of the big brown bat. So in the wild, we have a high percentage, upwards 90-95% of the colony is female. So when we're looking at hormone transfer from males to females, there's ample opportunity in a natural environment for hormone transfer to occur in bats here. In terms of reproduction and hormones in the big brown bat, we know relatively little, but compared to other bat species, we know quite a bit. So like other temperate bats, the big brown bat will mate in the fall. Females will store sperm throughout the winter, throughout hibernation. And if conditions are favorable, when the animal awakes at the end of hibernation, this is when ovulation, fertilization, and gestation is going to begin. So we have a decoupling of actual mating behavior and the time in which ovulation and fertilization is occurring. We also know that there is geographic and age-related patterns in bats. So in the West, big brown bats typically give birth to singlets, and in the East, we get twins. In terms of age, not a lot is known, but we do know that in the first year of life, when animals are juveniles or yearlings, they are reproductive to a lower degree than our adults. So in our colony, since I've been in the lab, we've had one female give birth in her first mating season, following her first mating season, the following spring. Literature shows that the same pattern is observed in the wild, with yearlings giving a lower rate of successful birthing. In terms of males, we know that our yearling males will mate with adult females. However, we don't track paternity in our colony, so we don't know if these males are actually fertile or if they're just mating for the fun of it, which is something I don't think we'll ever know unless we start doing some real genetic testing. Unknown aspects of bat reproduction include the hormonal control of reproduction. We know that luteinizing hormone and estradiol injections can stimulate ovulation, but other than that, we don't know much. We don't really know the mechanism behind this sperm storage. We know that it occurs reliably. It can occur for like hundreds of days. However, we don't know the mechanism behind it. Some people have speculated it might be a physical stimulation or hormonally based. And no one has really examined the potential of pheromones in bats. That's the work that we've been trying to establish. Should mention that big brown bats do have all the common mammalian hormones: testosterone, estrogen, progesterone, common reproductive steroids. So what I'm going to do for the remainder of the talk is walk you through 3 different projects that I've completed throughout my PhD, and looking basically on the physiological aspect of hormone transfer, and then into some of the behavioral work as well. And first, I want to focus on this element of hormone transfer from male to female bats. And the purpose of this current study is to look at seasonal differences in estradiol transfer. As I just told you, there is decoupling of mating behavior and ovulation and fertilization in the spring. And in the summer, there's no mating behavior at all. Bats reproduce once a year. They have one mating season, one reproductive season, and in the summer, it's strictly maternal. So we're going to look and see if there's differences amongst these seasons, because if estradiol is acting as a reproductive pheromone, we would expect it to this transfer to correspond to the times relevant to mating in the species. And in mice, we know that urine is a major vector for this transfer. So we're going to sample some of our male bats at these different seasons, and then again see whether the levels of estradiol in their urine correlates with the time of transfer. So in order to do this, we run a relatively simple paradigm in which we inject a male bat with radioactive estradiol. We cage him with 2 females for 48 hours, after which time we collect tissues from the females, dissolve those tissues, and analyze them using a liquid scintillation analyzer, and we were able to quantify the radioactivity in the female tissues. Now, we replicated this 3 times in each of the autumn, the spring, and the summer months. And what we see is that in higher levels of transfer is occurring during the spring and the autumn in comparison with the summer months. So the 2 aspects and 2 time points where the bat reproduction is important is the same times that we see the highest transfer of estradiol from male to female bats. I just want to point out a couple of tissues for you in particular, one being the hypothalamus, and where estradiol is known to stimulate both reproductive behavior and physiology in mammals, as well as the uterus and the ovaries, the reproductive tissues of the animal. And in these cases, we know that radioactively transferred estradiol is going to the reproductive tissues and binding at those locations. Now, we see variance in the transfer occurring between individuals and across time points. So not all of our tissues reach statistical significance after corrections for multiple comparisons, but the same general trend that higher levels of transfer are occurring during the reproductive months is there in all tissues, and in all cases we saw transfer from male individuals to female cohabitating individuals. So next aspect of this project was to look at the male urinary estradiol levels. And in order to do so, we go into our colony and we take all of our work tools. So we have a work surface of a trolley. We have wax paper to collect urine on. We can grab animals with our gloves and ID those animals using passive integrative transponders. So we can identify which animal we've actually collected urine from. We collect this in a hematocrit tube and we seal off the hematocrit tube. So we have all of our tools and we can label our urine. So we go into the colony, we're able to randomly select individuals to get urine from, hold those individuals, and you pray and hope that they're going to pee. Bats don't pee reliably, but it's a skill that I think I've gotten relatively good at, collecting urine from the bats. Never thought I'd have to go to postgraduate education to establish such a skill. It's a skill I think my parents are very proud of, that I've had to do a PhD in order to acquire, but hopefully that's a transferable skill for the future. They tell me about these transferable skills. I try to sell this one. Exactly. So we're able to collect this urine and we're label it and we freeze it until analysis at a later time, in which we use ELISAs, or enzyme-linked immunosorbent assays, to biochemically quantify the amount of estradiol in the urine. So these are our results. On the y-axis, we have the nanograms of estradiol per milliliter of urine, and we have the seasons on the x-axis. And what you see is that we don't have a significant difference in male raw estradiol levels across seasons. In fact, the summer actually has slightly higher estradiol presence. But when we deal with urinary quantification, we know that the hydration level of an animal can influence our readings. So in order to correct for this, we do creatinine corrections, and this allows us to correct for the hydration and activity level of an animal during the time of collection. So as you can imagine, during the summer, animals might be more dehydrated due to the heat than they would be in other seasons. And when we look at the creatinine-corrected estradiol levels, we see a significant difference between the spring and the summer and the autumn and the summer, in which the spring and autumn have higher levels of creatinine-corrected estradiol in the urine of these male individuals. So here we have the time in which estradiol is present in higher levels in a corrected quantity in male bats, corresponding to the same times that we see the higher levels of transfer occurring in our transfer studies during the reproductively relevant seasons. So, are there seasonal differences in transfer from male to female bats? Yes, the highest levels of transfer occurring during the spring and the summer. And is there estradiol present in the urine of male bats? And if so, are there seasonal differences? Yes, with in urinary adjusted levels, with the highest quantification occurring during the spring and the summer. So we wanted to go on and really focus on the urinary estradiol levels, the urinary steroid levels. And we wanted to establish whether these measurements are equivalent to that in the literature. Now, I was shocked, believe it or not. Researchers haven't focused on steroid levels in the urine of bats in the past. It doesn't seem to be as hot of a topic as I would have thought it would be. In fact, in big brown bats, nobody studies steroid levels really at all. And the few studies we have are in relation to castration of animals. So in this study, the castration or gonectomization of a bat dropped its androgen levels, steroid levels, drastically. You cut the animal's balls off and it doesn't produce androgens. This isn't a shock to anybody who works with animals or anybody that works in endocrine, but this is the type of work that's been done on steroid levels in bats. This is another study that just shows that with testosterone implants, you can bring levels back up to a significant level, and tracking steroid levels in female blood plasma following ovarectomies. So in order to quantify estradiol in urine, which hadn't been done before in bats, we went to validate these procedures using Uridols procedures of ELISAs that were validated and first created in Dennis Catanzaro's lab by Cam Muir. And what this does is allow us to quantify the levels of unconjugated bioactive estradiol in the urine reliably with very little cross-reactivity to other steroids and other forms of estrogen. So what we're measuring here, what we know, is actually bioactive estradiol in the urine. And these are the same procedures I used in the past study. But here, what we're doing is trying to do a large exploratory study. We're going to collect urine over one annual breeding season. That's a full year. Quantify urinary estradiol across this season and look at a number of differences. So we can look at sex differences with this data. We can look at age differences. As I said previously, we have less reproductive capabilities, it appears, in juvenile individuals or yearlings. And we can also look at seasonal fluctuations, again, in males with a larger sample size, as well as females. So what we do, again, is go into the colony, we collect our urine, and we analyze the urine using ELISAs. And as Paul alluded to earlier, this is really when the P came into my PhD many, many years ago with hundreds of samples. So what I'm going to do is always present to you, because this is urinary data, the raw estradiol nanograms per mil of urine, as well as creatinine corrected graphs. So I'm going to present to you both quantifications of urine. The sample size of each group is in the number in each bar. And in this case, we have sex on the x-axis. The gold coloring is adults, and the blue are yearlings. So first, we look at the figure on the left. We can see that in females, we have no effect on the raw estradiol levels of age, of either yearlings versus adults, and we don't see any effect in males either. Across sexes, we have no differences as well. On the right here, when we look at the creatinine-corrected estradiol levels, first we can see that we do have a sex difference between adult males and adult females, with adult males having higher levels of estradiol corrected for creatinine in their urine in comparison to females. And when we look at males, we also see that there is an effective age, with adults having higher levels. If we move on and look at seasonal effects, first in males, on the left, when we look at adult males in particular, we can see that the quantity of estradiol in the summer month in the green bar differs statistically from that in the spring, autumn, and the winter. We do not see this same pattern in the yearlings of males. And when we look at creatinine-corrected estradiol, we do not see any statistically differences in any of the adult or yearling groups. Now, if you look at females, the first thing you'll notice is we don't separate them by age here. That's because if you recall a couple of graphs ago, I showed you that there are no age differences in the estradiol levels of juveniles and yearlings. So when we pull them together here, we see a number of differences. First, we see that the spring differs from the summer and the autumn and the winter in raw estradiol levels. We also have differences in the summer to the autumn and from the autumn to the winter. Looking at creatinine adjusted levels, we have differences in the spring from the autumn and the winter, as well as the summer to the autumn and the winter as well. So through this long time scale, 1 year evaluation of urinary levels of estradiol in bats, do we see sex differences? Yes, we see adjusted estradiol level differences in adult bats only, so between males and females that are adults. Do we see age differences? Yes, we see adjusted estradiol levels, again differing in male bats between adults and yearlings. And in terms of seasonal fluctuations, we see a different pattern with a larger sample size than we saw in our initial study. Here we see adjusted estradiol levels differing in males during the summer from the other months, and adjusted and unadjusted estradiol levels differing through a number of seasons in female individuals. So throughout the time working with urine here, we've really been starting to think about behavioral aspects of and the behavioral implications of steroids in bats. And in the midst of all this, a paper came out looking at the effects of steroids in mice in a pest control type of an environment. So what I'm going to do here is picture two pest control boxes. On the left, we have an experimental condition. On the right, we have a control condition. And they both contain male pheromones and food bait. However, the experimental condition contains testosterone dissolved in ether, and the control condition has only ether in it. And what we see in this study is that these pest control boxes, the ones with testosterone present, caught significantly more adult females and juvenile females than did in the control group, or the control bait box. And we don't see any effect of either adult or juvenile males in this case. However, if we run the same study again, and this time replace testosterone with progesterone and estrogen mixes, we now see that we catch more adult males and juvenile males in these bait boxes than we do in the control. And this does not hold true for females. So there seems to be a behavioral effect in mice in this pest control study with the effects of steroids and the steroids present in the environment. Now, if we start looking at effects of odorants and smell and environmental chemicals in mating systems in bats, we see one of the greatest examples of this to be the greater sac-wing bats. And these are really cool little bats that have flaps of skin on their upper arm, and in which males will urinate and put saliva into and glandular secretions. And then they will go and waft this perfume that they've created at the females in order to attract the females. Individuals have different perfume compositions, but the females are able to tell the individuals who are reproductive and ready to mate just from their perfume blend. So this is what you can see here is a greater sac-wing bat coming up. He's displaying his perfume to the female. And the female there smells it. It looks like she might be taking some breaths of air in to try to expose herself more to this perfume. We also see scent cues present more recently in papers looking at fringe-lip bats, in which males have this odorous crust on their forearm. And this odorous crust corresponds to testy size, to a larger testy size, as well as to the production of glandular secretions that are specific in adult males. So only reproductive males have this crust on them. They form it themselves using a mix of saliva and, again, glandular secretions, and they put it on their forearm, and this odorous crust can therefore tell females that this is a reproductive male. So we started going down this pathway, trying to use urine from our bats to establish whether there are behavioral implications from the secretions. So the first question is, does urine act as an attractant in bats? And if so, do bats stay in the proximity of the urine afterwards? So to do this, we established behavioral Y-maze tests in our lab, widely used in the literature, but not as commonly used in the bat community. And the bats are able to crawl through the Y-maze here towards the urine of either male or female bats. So we conducted these experiments during the late fall, during the bats' mating seasons, and we did them at an ecologically valid time of 8 p.m. to midnight, when the animals are most active. So what we did is we bring our animals down one day before the experiment to acclimatize them to the lab from the colony. And then 5 to 6 hours before the experiment, we isolate these animals in individual cages. They have no food or water during this time, so they have no scent cues in their cage before going through these experiments. So this is an aerial view of our maze. We have a start arm or a neutral arm in which the individual starts at the end of the maze. We have two stimuli arms. They both will contain urine on filter paper at the end of the maze. The end of our Y mazes have wire mesh grids. So we're able to put fans outside of the mazes that are able to just gently blow the scent or any scents or chemicals towards the junction point of the arms. And to take you through a brief trial, what we do is first assign urine randomly to one of the two arms. And we place the urine on the filter paper at a portion of the arm closest to the junction. We use 10 microliters of urine. We drag the filter paper to the end of the arms to create somewhat of a scent trail. Introduce the animal to the maze and allow them to explore for 5 minutes, which is time that we settled on based on pilot data. And we film this using a remote control video camera, and then we can later go back and analyze the videos. So to give you an example of what this looks like in the lab, this video is sped up to 5 times the speed. What we can see is an individual walking up, following essential, investigating the filter paper. It poops, as an animal does in an experiment. It follows the trail, kind of goes over to the other arm, investigates the filter paper there, looks for a conspecific, can't find one, keeps following the urine trail and goes back and forth between the two arms. Now this is a particularly active individual, I will point out, but you can get the sense of what the animals are doing in the maze. From our videos, we're able to look at a number of variables. We look at the first choice that the animal makes. Do they first go to the male urine or the female urine? We look at the last choice they've made. So where is the animal located at the end of a 5-minute trial? And we also use 3 methods to quantify animal preference of urine. So we use an absolute time difference between the time spent between the male and the female arms, which we call the preferred arm. And then we also use 30-second thresholds and 60-second thresholds of the number of animals that spent at least 30 seconds more in one arm or a minute longer in one arm. So this data here is for female individuals. And we see that females preferentially go towards male urine when they're introduced into these Y mazes. So we have 12 females that went to the male urine, 2 went to female urine first, and we had 1 female who did not cooperate with our study and never left the neutral arm. When we look at the last choice, again, we see that females, we have 7 that stayed in with the arm of the male urine, 1 in with the female urine, and 7 that were in the neutral arm at the end of our experiments. So we had 6 that returned to the neutral arm following urine investigation. In terms of absolute time, females tended to spend more time with male urine than they did female urine. And the same pattern holds true with 30-second and 60-second thresholds, although they don't reach statistical significance. Now, if we look at the male behavior, we get a little bit different results. First thing you can see is that males have no choice if they go to male urine or female urine first. You also notice we have a larger sample size of 19 animals because we're just trying to get the males to cooperate. We had 9 males who never left the neutral arm, many of them never leaving the starting position. Evidently, they didn't read the research protocols before and didn't know what they were supposed to be doing. But we have a lot of stubborn males here. Now, this seems like a lot. 50% of males are not actually leaving the neutral arm. However, this percentage seems to correspond to other behavioral tests that we've run in our lab. And we just find that males are stubborn and won't go through a Y maze. They don't care to explore as much. In terms of last choice, we see absolutely no preference in the area in which the animals are spending time. And in terms of absolute time, we see absolutely no trends or differences in the stimuli arms the males spend time in. As I said before, the video I showed is a particularly active individual, and we have a lot of variability in individuals' behaviors. So here, animal 7 is a female who first went to female urine, spent a lot of time in the arm containing female urine. She might have left this arm and come back in, but she never investigated the male urine and ended with the female urine. Animal 13 is a female who first went to the male urine, did explore the female urine at some point, but returned to the male urine by the end of the study or the end of the trial. Animal 34 is a male who first went to the male urine, but investigated both male and female urine with relatively equal time and returned to the neutral arm at the end. And then here we see in animal 26, a male who just wasn't cooperating and never went anywhere except in the neutral arm and likely didn't leave that start position. So does urine act as an attractant in bats? We think yes, male urine appears to attract female animals. And do the animals stay in the proximity of urine after being attracted to it? And here we're kind of up in the air. We have trends going towards statistical significance of females staying in the proximity of male urine, but they don't reach our criteria for statistical cutoffs. So these 3 projects that I've kind of outlined for you today have a number of implications in the mammalian and bat literature. First, they highly support the notion that estradiol may act as a pheromone in bats. We have reproductive relevant transfer from males to females during those reproductive seasons. We have estradiol present in the urine of animals during all seasons. And we have different season fluctuations depending on the sex of the individual. And we've shown that there is some behavioral implication of urine in attracting behaviors of females to male urine. So with this transferred data, we like to hypothesize this is likely more of a mammalian-wide phenomena of hormone transfer, as we have now demonstrated it reliably and repeatedly in bats, and it has also been shown reliably and repeatedly in mice as well, 2 evolutionarily distinct species. And in doing so, males might actively be influencing the reproductive states and behavior of cohabitating females. This also has implications for hormonal control or possible regulation in bats. It might be, males might be able to induce ovulation in females or induce mating behavior, as well as possible mechanisms to induce ovulation, as I just said, as well as for sperm storage in bats, which is largely unknown to date, the mechanism of how that's occurring. Now, this is really just the start of this line of research, and there's a number of future directions in all aspects of these projects that we could look at in the future. And the first is estradiol transfer in bats through seminal fluids. DeCanzaro and Pollock showed previously that radioactive estradiol transfers in mice throughout mating directly to the reproductive tissues, and that estradiol is present in the seminal fluids of male mice. Estradiol has also been measured in seminal fluids and ejaculate of boars, horses, and many small mammals, as well as humans and other primates. So it is likely that this estradiol is present in the ejaculate of male bats. It just hasn't been measured to date. I also think we can look at behavioral observations when we're looking at this estradiol transfer. We do have variance between the amount of radioactivity present in the tissues of multiple females who have cohabitated with the same male. So whether that male is roosting above one female in particular or has mated with one female in particular is something that we need behavioral videos in order to analyze, to account for that variance. also think largely bat researchers should start looking more at the potential for Bruce and Vandenberg effects to occur in bats. As I said, the decoupling of mating and ovulation and fertilization makes some of these very hard to account for. Currently, we don't have a good mechanism to know when an animal has ovulated or if fertilization and implantation has occurred in a bat until you're able to actually palpate a fetus in the individual. And in knowing after an animal has mated, even we don't know, there are sperm plugs in some species of bats. Sometimes they're formed by males, sometimes they're formed by females, depending on the species. But they're too far internal in many species to be seen visually from the outside. So we can't just pick up a bat and say, yes, you've mated, no, you haven't, like we can with the mice. And lastly, I think we should take approaches much like Kakix et al. Study, looking at the effect of specific steroid combinations as attractants in bats. They looked at it in mice, and in our current study on bats, we've just looked at urine as a whole. And there's a number of other factors and proteins in urine that could be acting as attractants. So I just want to acknowledge everyone who's worked on these projects over the years. First, my colleague and friend, Tyler Pollock, who many years ago was more than willing to train me in all the radioactive protocols, how to process tissues, how to run ELISAs effectively and efficiently, how to clean when you use radioactivity. There's a ton of cleaning involved. And Tyler made sure that I was the #1 cleaner out there. I need to thank Dennis Canzaro and his lab for opening the lab and welcoming me with open arms when we started this collaboration between the bat lab and his lab, and being open to continuing this work in bats, giving me open access to his lab and resources, and continuing to do so as a professor emeriti. Joe Salter and Audrey Tam are undergrads in the lab. Were undergrads, they both graduated now. Joe spent a lot of time multiple years ago working on estradiol transfer projects with me. And Audrey Tam had, I don't know what I did, sorry. But Audrey Tam was essential in helping bring the behavioral testing to our lab and helping go through the literature and the bat literature and come up with established protocols for that testing. Thank my PhD committee, Joanna Wilson, Lauren Foster, and Aisha Khan, for all their support and advice throughout my PhD. I've probably sat on one too many committees and taken on one too many responsibilities during my degree, and they've always been more than supportive of that. As well as all the current and past members of the BAT Lab, you've all helped me and supported me throughout this work. Even if we haven't worked directly together on a research project, you've always been willing to lend a helping hand whenever I needed one, or bounce ideas off of, or ears. And of course, with the BAT Lab there, we always include our CAF staff, and Dawn, who always is more than helpful in looking after our animals and allowing us to keep the amazing resource of bats that we have. So all this work was funded by NSERC. And lastly, I just want to thank all my PNB friends for their support throughout graduate school, and for coming out today to hear about what I've actually been doing for the last 6 years. And last but not least, the man in the funny hat, Paul for always reminding us we always have to have fun in science, and to always keep an excited and approachable outlook on projects in project diversity and where that project's going to be leading in the future, and to follow the data there. Another lesson that Dennis has always told me and instilled on me. So thank you all for coming out today. I hope you've enjoyed the talk, and I'm welcome any questions you have about the data. Thank you.
Questions. There's also wine and cheese upstairs right afterwards. I think that was a nice example of PNB student research. Some of the studies P, neurobehavioral and physiology, and of course the behavior. Jim.
Lucas, I'm just trying to imagine bats giving birth and whether they would be in the proximity of the potential father when they give birth. And I'm wondering about the possibility of cannibalism and something that might drive the Bruce effect.
Yeah, so being in proximity, first of all, the animals aren't necessarily in proximity of the father who sired the offspring. Cannibalism can occur. We have had maternal cannibalism happen after birthing in our colony. We know that it does happen in the wild as well. In terms of paternal cannibalism, I think it's relatively unlikely just due to animals roost in different areas. They might not necessarily stay in the same caves during hibernation period or the same roost during hibernation. So in terms of paternal cannibalism, not really anything that I've ever read about or been aware about in bats.
I guess I'm wondering if that's the case, what would drive females? Why would you suspect the first effect would happen?
So yeah, okay. So it would be occurring before development of the individual. So although bats mainly mate in the fall, you do get occurrences of mating occurring in the spring and throughout hibernation. So males will even go around during hibernation and mate with a torpid female who's hibernating. Whether this is some kind of alternative reproductive strategy, we don't know. But males will mate in the spring as well with females, just at a much lower frequency. So you could get possible disruption of pregnancy in the spring, at what time a male is actually reproducing with the female immediately after or almost immediately afterwards, and then she can sire his offspring as well. So it's less, that mating occurs less frequently, but it does occur in the spring. Does that make sense? Kind of?
I still don't see the advantage of the female having a physiology to abort.
So it's really from a male perspective. The male is able to have the female sire his offspring as opposed to the offspring of another male. From the female perspective, if that male is going to stick around in the roost until later into the spring, then he could potentially avoid cannibalization or infanticide. But in our species, that's not as big of a concern. Yeah, it's more having the female sire his offspring as opposed to someone else's, as opposed to looking at the infanticide route.
I was just wondering if you can comment on like the uptake of these hormones by females and the response of the endogenous hormones to the exogenous hormones that you gain from via the male bat. Is that something that's passively happening? And when the preference that you did with the Y-mate, is that something that's passive that's happening, or is there some sort of a preference where females might react better to certain urines and not? So I'm saying, is all urine the same, or do females preferentially go towards some urine and block out other urine?
Yeah, we haven't gone that far into testing. All the urine in our behavioral tests are all from adult individuals. Each individual that urine was collected from, one male was exposed to that urine and one female was exposed to that urine, but we didn't have replicates from specific individuals, urine from specific individuals retested. So we don't know if there's like a dominant male that everyone's attracted to or a subordinate male that maybe someone wouldn't be attracted to. So that kind of level of detail hasn't been evaluated yet.
So there's some evidence that like some hormones can also be repellents. Is there no possibility that the urine is repelling the males and that's why they're staying in the same zone?
Yeah, just a different perspective on the data. There could be something in there that I wouldn't say maybe repels the males in particular because they don't show any sex differences. So if there was something repelling from male urine, you would think that the males would still go to the female. Maybe there's something in female urine repelling other females. But we do know that, so in the sackwing bats, there's greater sackwing bats and lesser sackwing bats, because bat biologists are that creative in our naming systems. And they cohabitate together, but it's the female who decides who to mate with, and it's the female who selectively goes to the animal for same species. So we know that females do use scent cues and chemical cues to make sure that they are mating with their own species. So similarly here, this could be something where females are particularly attracted to the urine of males of the same species here, and it's another female preference and choice in mating. Females are typically dominant in big brown bats as well.
Hey Lucas, great talking. So I was curious about what is the difference between torpor and hibernation, and how do you extract pee from individuals when they're in torpor or hibernation?
Right. So hibernation is kind of like long-term torpor. Torpor is just the metabolic state more or less. Hibernation is this extended torpor. Animals will arouse periodically throughout hibernation. That's normal. How do you collect urine? Very patiently. So when we go into the colony, typically what you're doing is you are disturbing an animal from their torpor state. You have to hold the animal for a while. You kind of rub their back. You try to get them warmed up. And once the animal is warmed up, that's when they're actually urinating. So it's a much longer procedure in the winter. And you are disrupting the animal from their natural torpor and hibernation cycle. So, yeah.
Any other questions? Well, if not, then please thank Lucas for a wonderful talk. We're all going upstairs to 205 for wine and cheese.